The nucleus is often referred to as the control center of the cell, since this structure ultimately directs the cell's many activities. The nucleus contains the macromolecule vital to all life, deoxyribonucleic acid, or DNA. The nucleus is surrounded by a nuclear membrane, which is composed of a double layer of lipids and proteins. The nuclear membrane controls access to the instructions encoded within the DNA. The DNA found in the nucleus contains coded messages called genes that each cell uses to make thousands of different proteins, enabling it to live and to reproduce. The instructions encoded along the DNA are eventually passed on to successive generations of cells as each parent cell divides. When cells begin to divide, the chromatin, containing DNA and protein, condenses and forms rod-shaped bodies called chromosomes. Chromosomes are responsible for carrying the hereditary information from one generation to the next. Within the cell, but exterior to the nucleus, is the cytoplasm. This jelly-like substance provides a viscous environment for all of the intracellular components. Over the last several years, scientists have learned that the cytoplasm is not a simple clear liquid inside the cell, but rather a highly complex solution composed of water, proteins, carbohydrates, and lipids. In fact, the cytoplasm is as distinctive as all the other organelles in the cell. Many chemical reactions critical to normal cell function occur in the cytoplasm. The cytoplasm is in motion, moving materials such as nutrients from one part of the cell to another. In large plant cells, this motion, referred to as cytoplasmic streaming, is especially common. Here you see the cytoplasm moving around the cell in the space between the vacuole and the plasma membrane. One of the most important and complex structures of the cell is its outer boundary, known as the cell or plasma membrane. The cell membrane serves two main functions. It protects the internal parts of the cell, and it regulates which substances enter and exit. The cell membrane is selectively permeable, restricting the flow of traffic into and out of the cell to specific molecules. It, in essence, acts as a gatekeeper. The cell membrane consists of phospholipid molecules and proteins. A closer look at a phospholipid molecule reveals that it is composed of a hydrophilic or water-seeking head and a hydrophobic or water-repelling tail. The phospholipid molecules that make up the cell membrane are in constant motion. Proteins that help make up the cell membrane perform several important tasks, including transporting molecules into and out of the cell. Some proteins act as small pumps, actively transporting molecules from one side of the membrane to the other. Just as most organisms are composed of many different cells working together, individual cells are made of numerous organelles that work in concert to ensure that the organism functions properly. The transportation of nutrients, wastes, and cell products within the cell is vital to its ability to function normally. Such transportation is facilitated by the endoplasmic reticulum, or ER, which forms an interconnected highway system on which molecules can move from one part of the cell to another. In electron micrographs, the ER appears as loose strings running throughout the cytoplasm. These structures are actually delicate membranes of sacs and tunnels. There are two different types of ER. One type, called rough endoplasmic reticulum, appears to be covered with tiny spheres. Rough endoplasmic reticulum extends from the nucleus and is indeed covered with small structures called ribosomes, which are responsible for the rough appearance. The rough ER connects to the nucleus and is important in transporting certain substances, such as proteins, into and out of the nucleus. Cells that produce large numbers of proteins have more rough ER than those that produce fewer proteins. In addition to providing a surface area for ribosomes, rough ER serves as a storage area for newly synthesized molecules. Another type, called smooth endoplasmic reticulum, has no ribosomes attached to its surface, and thus appears much smoother than rough ER. Like rough ER, smooth endoplasmic reticulum serves as a storage area, mainly for proteins that will later be exported out of the cell. Ribosomes are the most numerous structures in the cell that may or may not be attached to the rough endoplasmic reticulum. Ribosomes, which are composed of RNA and proteins, are the primary site of protein synthesis. Since all living things rely on proteins for cell maintenance and growth, it is easy to understand why ribosomes are so abundant. The location of ribosomes within a cell provides an indication as to their specific functions. Those located on the rough endoplasmic reticulum synthesize proteins that will eventually be used in the cell's plasma membrane or exported out of the cell. Ribosomes that float freely in the cytoplasm synthesize proteins that will be used inside the cell. Every eukaryotic cell has a uniquely shaped organelle called the Golgi apparatus. Here you see the structure of the Golgi apparatus, which appears as flattened sacs stacked on top of each other. The Golgi apparatus sorts, processes, packages, and delivers proteins and lipids throughout the cell. The proteins and lipids may remain inside the cell, be used in the plasma membrane, or be exported outside the cell. Thus, cells that produce high quantities of proteins and lipids have a very extensive Golgi apparatus. Lysosomes are membrane-enclosed vesicles that form in the Golgi apparatus. They may contain over 40 different powerful enzymes that enable the cell to digest and destroy large molecules. Lysosomes help white blood cells destroy foreign substances, such as viruses and bacteria that invade the body. Lysosomes also recycle older or damaged organelles. By engulfing selected organelles and digesting them with powerful enzymes, lysosomes return the digested components to the cytoplasm, where they can be reused. This process helps the cell to continually renew itself. Some organisms use lysosomes to break down food molecules that have been engulfed by their cells. For example, amoebas eat by engulfing small prey or food particles. Once inside the cell, small food vacuoles containing the captured substance fuse with lysosomes, which release their digestive enzymes. You may have heard the mitochondrion referred to as the powerhouse of the cell. This important organelle generates energy in the form of ATP to keep the cell alive. Some cells may have one large mitochondrion. More often, however, cells contain hundreds or even thousands of mitochondria. Cells that require more energy, for example, kidney, liver, and muscle cells, have many more mitochondria than cells that require less energy. Structurally, mitochondria vary in size and shape. However, all mitochondria have two membranes, an outer membrane, which is smooth, and an inner membrane, which is arranged in a series of internal folds called cristae. The many folds of cristae provide an enormous surface area where vital chemical reactions occur. This chemical activity in turn provides energy for the cell. Mitochondria are semi-autonomous, that is, they can grow and self-replicate within the cell. Each mitochondrion contains its own ribosomes and a small amount of DNA. Interestingly, mitochondria resemble bacteria in size and biochemistry, and some scientists believe that eukaryotic mitochondria evolved from ancient bacteria.